Welcome to the Beer Taster's Notebook, the show focused on beer education. I'm Jonathan Thompson, Advanced Cicerone, BJCP beer judge, and fermentation enthusiast. Welcome back to the Beer Taster's Notebook, episode 23, Yeast Reproduction and Metabolism. So we just talked about, yeast in general last time. I'm really splitting this yeast series up into a lot of different parts just for navigational purposes and because it's a lot of information. So today we're going to talk about the birds and the bees of yeast, or maybe just the birds, or maybe just the bees, or maybe neither. I'm not really sure. So most of the time, brewer's yeast at least reproduces asexually. So that's why I'm alluding to no bees, no birds, lots of bees, lots of birds, whatever. So asexual production, or reproduction rather, can take the form of budding, where the cell wall kind of bulges out into a new cell and then breaks off. Fission, where cells divide into two cells, and then sporulation, where the parent cell makes tiny little special clone cells called spores that are then dispersed. So fission is common with bacteria, sporulation with many fungi and some plants and some bacteria. So if you think about a mushroom, it's the fruiting body of the fungus, and then it emits these spores, and they sort of fly around all over the place. They're very light, so they can be carried on insects. They can be carried by air, and then that's how the species disperses itself across the universe. And then, so budding is the most common form of asexual reproduction in yeast, at least in brewing yeast. So yeast can exist in two forms, known as diploid and haploid. Diploid, like human cells, contain two copies of each chromosome, and haploid only carries one copy. Humans, by the way, have 23 chromosomes, and yeast have 16, just for a little cocktail party knowledge there. So wild yeast is usually diploid and has the ability to sporulate. This is good from an evolutionary perspective and creates much needed diversity. And as we know, chromosomes contain the genetic material, and so yeast, by the way, have about 6,000 genes. And so through selective pressures, brewing yeast are no longer able to sporulate or really even to mate. And so they seem to have become polyploidal, which means they have many copies of each chromosome. And why this is important? Well, okay, so if let's say you write me a note that says buy beer, and then I write this down and hand it to someone else, but my handwriting's terrible, which is true, and the R looks like a T, and then either the note goes down the line saying buy beets and becomes unadulterated, or even worse, their T starts to look like an F, and then we get obviously something that we really didn't want in the first place. And so maybe that's not a very good analogy, but I think it captures the spirit of it. So if there are two copies of each, then the error is less likely to make it through, and so on. If you're handing down multiple copies, then the error is basically completely eliminated. And so let's talk about budding now. So we'll focus on budding as the reproduction method for this episode, as it's the most important to beer. We can look at sporulation and fission and such things down the road, especially when we start talking about other organisms. But first, let's talk about the yeast cell itself. Well, I guess just the yeast, it is a single cell, but either way. Okay, so let's just say the structure of the cell. So the yeast cell is spherical or ovoid in shape generally. I mean, they can become a little bit elongated and abnormally shaped, but those are the general shapes. Remember, it's a eukaryote, so unlike bacteria. And so the cell contains a nucleus, mitochondria, a Golgi apparatus, vacuole, ribosomes, and this is all contained in a cytoplasm and bound by a plasma membrane around which is wrapped the cell wall. So the cell wall is made of chitin, and this is the same substance that comprises the exoskeletons of arthropods, like crabs, and also it's the same thing that sort of coats the outside of a mushroom, which makes parts of mushrooms sort of hard to digest, so people say. I mean, I don't really have any issues with that, but anyways. Manoproteins, which are a glycoprotein, which means it's a sugar and a protein, and then beta-glucans. Beta-glucans are a soluble fiber, and they're found in abundance in oatmeal, which is probably where you've heard that. And so just a bit of cellular physiology. So the cell wall, I'm sure you can guess, keeps stuff in and out, and then provides structure. The periplasmic membrane, which resides between the cell wall and the cytoplasmic membrane, which I will get to next, is where the enzymes hang out. And the cytoplasmic, why can't I say this word? Cytoplasmic membrane allows nutrients in and metabolic waste out. So it's kind of like the gateway in and out. And so cytoplasm is, it's kind of a grainy matrix, but it's probably easier to just think about it like a gel-like fluid. And this is where metabolic events happen. So The nucleus, which is also membrane bound, is the command center. This is where genetic material is housed and where metabolic systems, cell growth, reproduction, et cetera, are all controlled. Ribosomes are little molecules that synthesize protein from the instructions that they're getting from the RNA, which is coming, of course, from the nucleus. And the Golgi apparatus, it's the way that it's often described is like a shipping warehouse. So proteins and lipids and such things are processed, packaged, and then sort of sent to where they need to be, if that makes sense. So it's kind of like it distributes various proteins and enzymes to the right location, including out of the cell. I mean, things that need to get pushed out will be pushed out by the Golgi apparatus, or at least packaged and sent in the right direction. And then the vacuole is where nutrients are stored. And then mitochondria, that's the energy center. And this is where respiration occurs. This is where the Krebs cycle and all that kind of stuff occurs. So that's where the energy is generated. Okay, so now back to just budding. So the process here kind of looks like this. A little bubble appears. And if you think about like a stress ball, one of those gel-filled ones, or even like if you had a basketball and you got a weak area on the wall, and then you get this little bulge that grows out and it becomes sort of abnormally shaped. That's exactly the same sort of thing. It's almost like a weak spot in the cell wall. Of course, in this case, it's on purpose, not just a cheap basketball. It grows, and the parent cell's nucleus divides into two, and one of those divisions goes into this bud, or this budding. Other organelles, such as the mitochondria and vacuole and such things, will undergo division as well, with one of those going into the bud. So we basically get a complete copy stuck in this bulge. And then once mature enough, the bud will pinch off and it goes out on their own. And so this bud is called a daughter cell, and then the mother cell can just do this again, usually 20 to 30 times, ideally. And this takes about 90 minutes per budding. It leaves a scar, called a budding scar on the yeast. And this reproduction, as mentioned, offers very little genetic diversity. And this is very key for creating the same fermentation conditions over and over again. And so this consistency obviously is very important to brewers. And if there were significant or really even insignificant genetic variation from one generation to the next, we'd have zero idea what was going to happen in the fermenter. And so this might be pretty fun, but it's going to be a massive issue for the average consumer and ergo the brewer. If you're somebody who wants to experiment or likes experimental beers and likes to taste different things all the time and doesn't expect a consistent product or even want one, then that's probably, wild yeast is the way to go. But if you're somebody who wants their beer to taste exactly the same every time, especially people who just and you're probably not listening to this podcast if that's your kind of thing. And if it is, it's none of my business. But if you're somebody who buys the same beer over and over and over again and only drinks that one beer, and you might buy it in a case of 2, 4, or whatever, you're probably not going to want variation. And the massive brewers and even microbrewers, really, they don't want much variation either, right? They want, if you want, go into a place and actually, you know what? Here's a better example. So that I don't take the risk of insulting anybody, I want Guinness to be exactly the same everywhere that I drink it. And so there you go. So I'm putting myself in the same category with everyone else. And so if they didn't have proper QA and they didn't have their yeast sequence and all the rest, and they had all this variation, we would never know what the hell we were going to get. And everyone would be upset, including myself. Maybe I wouldn't be upset, but I would probably be a little confused. I've got off track here. Okay. So the observable qualities of any organism, but the yeast in which we're speaking is called a phenotype. And so things like flavor expression, fermentation characteristics, like what temperatures they can tolerate, what conditions they are ideal at, that's called phenotype. The genotype is the genetic map. The yeast actually the genome has been sequenced. It was the very first eukaryotic organism to undergo such sequencing, but phenotyping is a little less onerous. Most brewers are not going to be, or even yeast labs. Well, I guess yeast labs may be different, but they're not going to be, genotyping or mapping out the whole genetic structure of every single one of their yeast strains. So that's probably as much as we need to really talk about with reproduction. So let's talk a little bit about food and metabolism specifically, of course. But so yeast, like everything else, needs to eat or at least derive energy from something. So plants, of course, use sunlight and convert that into energy with the help of carbon dioxide, water, and various nutrients. Yeast are a little further up the chain in that they're chemoheterotrophs. So this means they need to get energy from other compounds. And so those compounds are primarily sugars. And like everything, they want to gain the most from the least. So this means the biggest energy gain with the least expenditure. So yeast will eat the simplest sugars first. Glucose, then fructose, then maltose, then maltotriose. And so the yeast also need nitrogen to synthesize amino acids and build proteins. You can listen to that whole fan episode for more on that, but we'll still touch on it here. And then some other nutrients, including iron, zinc, calcium, and magnesium, as well as sulfur and phosphorus are needed. Zinc is usually the one that seems to be sort of scarce, and sometimes we need to add that in. Zinc, by the way, is essential for humans as well. And the enzyme that converts the acetaldehyde into alcohol in the fermentation process is basically the same enzyme that converts alcohol to acetaldehyde when we digest it. Ergo, if you're drinking beer, take your zinc. I got lost. Did I? No, I didn't. Okay, so the yeast also needs lipids. Those are found in wort, but a lot of it they have to synthesize. There's not a lot of lipids are fats, of course, but there's not a lot of them floating around in your wort. And if there were, then you'd have terrible foam. And so it's kind of a tug of war there. But most of the time they need to synthesize these fatty acids themselves. Okay, so let's look at the fermentation process now from a human point of view. Because I think we covered a lot of fermentation and metabolism, so you can go back to that respiration fermentation episode just to get sort of to fill in here if you're a bit lost as to what's happening, at least at the fermentation stage. But this is from the human point of view. So we're looking into the fermenter here. So we've got wort and we've cooled it after we've boiled it. We've cooled it to the appropriate temperature and it's in the appropriate vessel. And so now we need to introduce yeast, obviously, to ferment it. I mean, we could leave it open and see what happens and get some wild yeast in there, and that could be cool, and that could also be very bad, and et cetera, et cetera. So just a side note, also, some dried yeast needs to be rehydrated. Some need to be boosted up and to sort of to get a head start, especially depending on the composition of the wort. And you can use a yeast starter for this, and we could talk about that separately. And we could also talk about oxygenation separately, because I think we could do a whole episode on oxygen. But just to touch on it here, yeast requires oxygen to synthesize sterols, which are integral for cell walls. And so, like I said, we'll deal with that later. So now we pitch the yeast into the well-oxygenated wort, and phase 1 begins. So phase 1 is often called the lag phase, because to the naked eye, nothing seems to be happening. To the fully dressed eye, if that's a thing, a lot happens. And so first, the yeast are going to undergo the process of acclimatization. They're just being dropped into a new environment with different temperatures, different conditions, especially different levels of oxygen. The pH is different. It's kind of like going to a new school or a new class, right? I mean, it's just you have to figure out who's who in the zoo. And so the yeast start taking up oxygen, nitrogenous compounds as well, and other various nutrients. And from these, they're building the amino acids and proteins they need, and the sterols, of course, and they're getting ready for stage 2. And so really importantly, they start repairing and reinforcing cell walls, which can undergo some stress during storage and sustain damage. So, just think about this, the yeast is going to start growing and budding. And if those walls aren't healthy and flexible and strong, well, it's just going to be bad. And the sterols, as mentioned, require oxygen for building, and they are very key to a healthy cell wall. And so then the third sort of major thing is that the growth pathways start being turned on and metabolism starts to kick in. So at this point, no saccharides, so no sugars are being ingested really. It's all oxygen, nitrogen, and then minerals and vitamins. But the yeast will grow in size, up to 41% or so. But there will be no bubbling or anything like that. There's very little to no CO2 being spit out, and so we're not going to see any action, at least on the surface. The pH of the wort will drop a bit, and there's several reasons for this. So that little bit of CO2 is in the form of carbonic acid, and so as much as this might be minute, it can contribute to a small drop. And then the action of proton pumps to facilitate the uptake of nutrients will release some hydrogen ions into the beer as well. And some nutrients like phosphates that were acting as a pH buffer in the wort are being taken in, so that shifts the gradient as well. And also some of the amino acid uptake results in the release of lactic and acetic acids. And so that's obviously going to change pH as well. And so this whole process can be pretty quick, but it usually typically takes around 15 hours or so. And then next comes the growth phase. And so this is pretty much exactly how it sounds. And when reproduction happens as well, it's also called the exponential growth phase or the log phase because the growth is exponential or logarithmic. And as anyone who has been or known someone who's pregnant, reproduction requires food and energy. And so this is also when the yeast start consuming all these sugars in the wort to produce more energy. And the metabolic products of this, of course, are alcoholic carbon dioxide, along with a bunch of byproducts. So the cell count will increase immensely. And so this activity becomes quite visible. It's also called high Kreuzen. The rampant carbon dioxide production is going to carry particulate to the top of the fermenter, and it makes this kind of yellowy or off-white bubbling mess with brown flecks, mostly from hop and malt particulate. But it's that big bubbly, foam. Let's call it foam on top of the fermenting beer, and so the pH is going to drop way more quickly at this point too, because carbonic acid is going to increase as well as all those acidic metabolic products from the nutrient uptake, and this is the stage where nutrients really matter. And so if oxygen was the star, say, of the lag phase, FAN and other trace nutrients are the star of this phase. And so this can last up to 4 days. It can be as little as a few hours, depending on conditions, depending on the yeast. I mean, there's a lot of factors that play, depending on, obviously, temperature, and depending on what's even in there. I mean, if there's not very much available sugars, then it's going to end pretty quickly because they're going to run out of stuff to eat. Okay, so the third stage is now the stationary phase. This is also called the post-dioxic or G0 phase. There's very little activity that we're going to be seeing. Most of this, most of the primary fermentation sort of is complete, and most of the flavors have been produced, but the beer is not in any way ready. So this phase, by the way, gets triggered by the scarcity of glucose and other nutrients. And basically the yeast ate most of everything on the table, and now they're awaiting a second course. Maybe they're picking away at the rapini, the stems, or the parts that, but there's bits and pieces of trace stuff that they're pulling back in and whatever, but the main meal is done. The cells stop dividing, the cell walls start to thicken, and the cells themselves shrink a little bit, and then flocculation begins. So flocculation, which probably deserves a standalone episode too, is when the cells all clump together and then drop out of suspension. And a few things happen on the metabolic side too here. So the yeast, and so they start building glycogen stores or energy stores, but it's mostly in the form of glycogen. And this is pretty similar to human beings. So if you go lift weights, run, chop wood, or do anything strenuous, for a while, you're going to deplete the glycogen, which is stored in your muscles and eventually your liver. And then if you come inside and have some honey or banana or bread or something like that, then the body's going to break these down into individual components as we do during digestion and metabolism. And then it's going to store this back into the muscle and liver as glycogen. So once those stores are full and we keep eating honey, well, that's when we start needing new pants and things like that happen. And then eventually we can push ourselves into metabolic stress, but that's a topic for a totally different podcast. So that basically what the yeast is doing is it's storing energy for a later time when there's absolutely nothing to eat. And so this is when we apply the term green beer at this stage as well. And so there are various flavor compounds that are kind of less than the desirable in the end product. And the yeast will now take those back in and transform them into something else. So it's kind of cleaning up the off flavors, so to speak. And this is why we do secondary fermentations, and this is kind of what lagering is. And so that we'll do a standalone episode on yeast off flavors and how they're produced and cleaned up and all the rest as well. Okay, so now that there's not really any sugar to eat and glycogen is full-ish, the yeast shifts out of fermentation and begins survival mode. Stored glycogen is used as sparingly as possible. And ethanol is taken back in and converted to acetaldehyde, which is converted to acetyl-CoA, which then enters the TCA cycle. And then we don't want this, of course, because we want that alcohol to stay in the beer. And some uptake is fine, because if we're pulling back some alcohols, especially fusel alcohols, and we are combining those and making esters, then we, of course, get these wonderful aromas that we love so much. You can go back and listen to the episode on esters for all you need to know on esters and esterification. So I think that I'm going to leave it there. If I talk about everything in this process, then we're going to be here for hours, and that's going to be not good for anybody, and no one will get any rest. So I think that what we'll do is go through some of the enzymatic processes for each of the sugars and nutrients, and then we can talk about oxygen and fatty acid synthesis in depth. And then, of course, as promised, off flavors. We can talk all about temperature. So there's lots of things to talk about, and I think that just for navigational purposes, we'll keep these to sort of shorter and unique episodes. I have no idea why I couldn't remember the word unique, but anyways, I will leave it there. Until next time, stay thirsty and stay curious.